Okay, it's time to go ahead and get started. Want to welcome everyone to our GCC Innovative Drug Discovery and Development Workshop. We're excited today, particularly to hear from two leaders with important perspectives in global SARS coronavirus 2 vaccine development strategies, and also what it'll take to actually make those vaccines accessible. Before we get started, I do want to cover a couple of items. First of all, your microphones should be muted throughout. Please keep those off. Also, if you have any questions, you can enter those into the chat. We're not going to discuss those until after Dr. Nozel's presentation, but speakers may respond in the chat if they see your question. As such, it would be really good if you could put the name of the person to which the question is addressed at the beginning of your question. I know that both are going to mention this again, but I want to state in the beginning that there may be some questions that the speakers are unable to address due to various confidentiality agreements, so please keep that in mind. With that, I'll turn this over to Dr. Stan Wadowich, who will be moderating questions and introducing our speakers. Stan?
Well, thank you, Suzanne. I want to first of all thank everyone for joining us. Want to share a little slide that I found. Alan is the gentleman with the arrow over his head in this picture. I hope everyone can see it. So Alan,
brother.
What's it? So I've known Alan for over 20 years now, when he was one of the founding members of UTMB's Dream Team. And 20 years ago, Alan was tasked and one of the leaders on trying to stop viruses that were emerging around the globe. And here we are in 2020, and Alan is still at it. He is director of the Seeley Center Institute for Vaccine Sciences at UTMB, professor of pathology, microbiology down at UTMB, the director of UTMB's collaborating Center for Vaccine Research with the World Health Organization, international fellow member for the Society of Vaccines, and a fellow of the American Academy of Microbiology. So Alan has an extensive track record looking at vaccines, developing vaccines, tackling numerous pathogens, and we're delighted that he has time in his very busy schedule, because he is busy looking at this pathogen, and I think he'll tell you that he wasn't planning on doing it, but you know, you can't keep a good man down. So he is right in the middle of the vaccine efforts, and he'll share some of the work that's been going on. And so I'll turn it over to Alan, and I'll mute my mic.
Thanks, Stan. Just let me get the screen up. Stan, is that you? Oh, it's mine. Okay, there's the slides. Let's see the slides. I don't see my slides. Where are they? Ah, there you go. So thank you very much, Stan, for inviting me to talk about COVID vaccines. And I'm going to spend the next 30 minutes really on a warp speed overview of where we are with COVID vaccine development. There's a lot to cover, and I'm going to try and cover as much as I can related to the questions. And I really appreciate the opportunity to talk about COVID vaccine research from a global perspective. I have to give a couple of conflicts of interest. As Stan said, I'm the director of the WHO Collaborating Center for Vaccine Research, Evaluation, Training for Emerging Infectious Diseases. So I'm actively involved in a number of emerging disease vaccines. I should say we're part of the Initiative for Vaccine Research, IVR, not emergency medicines. And just to make this point, the COVID research efforts at WHO are coordinated for emergency methods, outbreak investigations, not for IVR. And as Stan indicated, there are some things that I can't talk about. I'm also a member of the Scientific Advisory Committee for the Coalition for Epidemic Preparedness Innovation, CEPI, and CEPI is supporting 9 COVID vaccine candidates. So I do potentially have some COIs here. So let's start by looking at the genome of SARS-CoV-2. As with most coronaviruses, it has a large genome, just under 30,000 nucleotides. It has 14 open reading frames, and this encodes 27 proteins. The virus has 4 structural proteins, which are shown here on the cartoon in red, S, E, M, and N. And the one that's the most important is S, the spike protein. And in fact, nearly every vaccine candidate is focused on the spike protein in one form or another. So if we look at the spike protein, it has little genetic variation, at least at the level of vaccinologists. We're not worried really about the sequence variation seen so far as part of vaccine development in 2020. Or this may change in the future if the virus evolves. The S1 subunit of the spike protein facilitates binding of the receptor binding domain, or RBD, to the ACE2 receptor. The S2 subunit of the protein mediates membrane fusion. And importantly, neutralizing antibodies to the virus are induced by the receptor binding domain, the N-terminal domain, and the stalk regions. The RBD antibodies are thought to be the most potent, and that's why it's been the major target so far of investigation. In fact, some of the vaccine candidates just express the RBD rather than the entire spike protein. The spike protein exists in 2 forms called pre and post-fusion. And this is important here because stabilizing the pre-fusion form is important for the binding of antibodies to induce an immune response as compared to the post-fusion form. So there's been extensive work done trying to get that structure sorted out in terms of actually when you express the proteins and the vaccine candidates, it induces the appropriate immune response against the spike protein. Many groups are making human monoclonal antibodies with neutralizing activity from patients who have recovered from COVID, and this is a very exciting field and probably offers in the short term a good way to actually prevent therapeutically the disease because you can express monoclonal antibodies in cell culture easily, make large quantities easily, and it's the way forward. And there are a number of groups working on these human monoclonals. But as I said, these human monoclonals all map to the spike protein and predominantly to the RBD. The structure has been determined for the spike protein for the virus, and that was first done at the University of Texas at Austin. And this shows the structure of the protein together with the interaction of the receptor binding domain with the ACE2 receptor. So we know a lot of fundamental structure function about the protein, which is very important for vaccine development. Now, when we think of vaccine development pipeline, we think of the basic science discoveries that are done. This then leads to the preclinical testing and manufacturing, looking at immunogenicity, efficacy, and safety in animal models, together with what I call low-scale manufacturing to make the lots to do clinical evaluation. We then go through the phase 1, 2, 3 clinical trials, and then we actually license the vaccine and we implement it with public policy and public education. This is the classical pathway. The classical pathway takes 20 years, approximately over $1 billion. And of course, with COVID, we're trying to actually compress everything into a matter of months rather than years. Now, it's important to understand the development pipeline, a lot of it is regulated. And so the basic science discovery work is not regulated in the labs. But as soon as you start going into preclinical development, GLP becomes important. As you go into the clinical trials, it's all GCP. And when you go to manufacturing, it's GMP. And all this is required by the regulators to make sure that the vaccine candidate you're producing meets the standards that the FDA require. Now, what are those standards? And the FDA has some lovely terms for considerations of a vaccine for approval, for safety and efficacy. And they call safety the relative freedom from harmful effects, purity the relative freedom for extraneous matter, and efficacy is a rather vague term, reasonable expectation that when administered under adequate directions for use, will serve a clinically significant function in diagnosis, cure, mitigation, treatment, or prevention of disease. Essentially, we're trying to get a vaccine that is what I would call 100% effective, and of course, 150% safe. That is the expectations of society and what we're trying to do. And when we have Operation Warp Speed, as we have now, it is a challenge to keep up with everything. But what I want to reassure you during this talk is everything is being done to meet the standards one would expect from any vaccine candidate. There are no shortcuts in terms of safety and efficacy being taken here for COVID vaccines. It's just that everything is going at an incredibly fast pace. Now let's go back to the beginning for coronaviruses and look at the veterinary coronaviruses. There are a number of vaccines used in the veterinary world for coronaviruses. Coronaviruses cause respiratory and enteric disease in animals. There are vaccines for cattle, pigs, chickens, dogs, and cats against different coronaviruses. And trying to big picture summarize it in general, live vaccines work better than activated vaccines. And in general, immunity is short-lived, requires frequent boosting, and may not prevent reinfection. And so we could say that the vaccines are not very good in the veterinary world. And so there's the concern when we move to the human translation, we're going to have short-term immunity, we're going to require frequent boosters, and we may not prevent reinfection. These are unknowns at the moment, but there are really important considerations as we move forward with human vaccine development, because it has proved incredibly hard to make coronavirus vaccines. One of the arguments why the veterinary vaccines are not as good as what we're doing in the human vaccines is the purity of the veterinary vaccines. Our technology for making human vaccines and technologies are much, I can say, superior in terms of purity, and we're hoping that these vaccines will have much better immunogenicity and require less boosting as their veterinary counterparts. But this remains to be seen, but it does set the picture of where we stand currently with COVID vaccine development, at least in the veterinary world. Now, when we move into looking at human vaccine development, we need preclinical models to assess the vaccine candidates. And the preclinical models that are used at the moment are mice, hamsters, ferrets, and non-human primates. Mice come in two categories: wild-type mice and mice that have a knock-in of the human ACE2 receptor. And these are transgenics. Now, the transgenics are very attractive, but the current price of those is over $1,000 a mouse, which it makes it somewhat of a challenge to actually work with these with large numbers. People are now working to try and adapt SARS-2 strains to wild-type mice, but this is still in its early stages. The hamster model has proved to be a very useful model for many people. It gets clinical signs, which represent the disease, but however the animals recover. Ferrets tend to get a non-clinical disease, and we have non-human primates. The non-human primates, they vary by species in their response to the virus, with the African green monkey probably being the best model at the current time. The important points of the models here are: one, there's no lethal infection; two, the timeline for the infection is relatively short compared to what humans have. All the animal models get mild to moderate clinical signs, and of course the animals recover. And we have very, very limited information on biomarkers for protection. I'll talk more about those protection markers as we move forward, but it really is a very big gap in knowledge that we don't really understand the correlates of protection in terms of our animal models. Clearly, the critical point here is these animal models have to be used as validated models to test the drugs, antibody, and vaccine candidates. And there has to be the expectation that the data produced in these animal models may get reviewed by a regulator as part of the approval process moving forward. Let's look at correlates of protection. Correlate of protection is a marker of immune function that statistically correlates with protection after vaccination. And it may be either mechanistic correlate of protection or non-mechanistic, which does not cause protection, but nevertheless predicts whether this vaccine is going to work. And if we look at correlates of protection, the studies that are normally done to identify what will be a correlate are passive antibody administration, analysis of the immune responses in protected and unprotected humans, observations of immunosuppressed humans or animals, human challenge studies, and extrapolation from the animal model studies done in preclinical development. So if you look at passive antibody studies, there's some pretty good evidence with convalescent plasma that this actually will ameliorate the disease, suggesting there is something in the plasma, probably antibodies, that helps mediate protection. The immune responses in protected and non-protected individuals are currently about to start in efficacy studies, so we don't know much about that. Our studies on immunosuppressed humans and animals are somewhat observational. Human challenge studies is an area very active at the moment. The question is what we call CHIMs, controlled human infection models. Is this the way to go rather than large clinical trials? Do small studies with CHIMs, with volunteers willing to be immunized and then challenged with wild-type strains? There are lots of concerns about this, particularly ethically. And the major problem is we can't do what we call a rescue. That is to say, there is no drug available. If somebody had severe disease, we could give them to protect them against the disease. So a surrogate of protection is an immune marker that can substitute for a clinical endpoint and could reliably predict efficacy. And these are always very useful in terms of the potency of vaccines when they go towards licensure. So how do we define correlates of protection in terms of COVID? We could say it's neutralizing antibodies, lack of clinical disease, reduced lack of viral load, or is it going to be memory? And I would say at the current time, neutralizing antibodies are probably our best bets. They're a surrogate for many licensed vaccines. The data for COVID convalescent plasma looks pretty good, a question mark, but I think looks pretty good to date. There are issues about potentially other functional antibodies, such as inhibiting fusion, or just ELISA antibodies may play a role as an assay to measure protection, depending on the particular vaccine candidates. Lack of clinical disease is not an obvious one to do, nor is reduced viral load. The issue of memory and T cells is an interesting area, but really is a basic science discovery stage at the moment, and there is no evidence really, as far as I'm aware, indicates we want to go towards a T cell based vaccine. Now, the other side of the coin is something called disease enhancements. There are studies done in animal models that show coronaviruses can cause disease enhancements. And I want to emphasize this is something that is being shown in animal models for different coronaviruses, but is theoretical for the virus in humans. This has been studies that have been written up by CEPI and the Brighton Collaboration. The Brighton Collaboration is a working group that looks at vaccine safety, and they've done an evaluation of disease enhancement with respect to COVID. There are two potential hypotheses that are put forward to explain disease enhancement. One is antibody-dependent enhancement, and that is where either poorly neutralizing or non-neutralizing antibodies bind to the virus, make a virus antibody complex, and that complex is taken up via Fc receptors into monocytes or other related cells, and that can lead to enhanced disease. However, this has to be taken very carefully when you do the interpretation. You can take near enough any virus, put it in cell culture, and demonstrate antibody-dependent enhancement. For example, my favorite virus is yellow fever. We have a great vaccine for it, 17D. It's been given to 900 million people. I can take sera from vaccinees, put it in cell culture, and get antibody-dependent enhancement of infection. But there is no evidence that takes place in humans with 900 million people being immunized. So we have to be careful how we interpret ADE. I think more likely is an interesting studies where it's suggestive that it's actually the type of immune response induced by a vaccine that may be a potential problem. Dominant T helper Th2 type response to the vaccine antigens appears to be one that leads to immunopathology. It's not seen if you use vaccines that have adjuvants or other modulators in them that lead to a Th1 response. A complexity of this is something going back to the veterinary vaccines. The purity of the vaccine may have a role as well. There may be some confounding effects of some molecules, proteins, or something else in the vaccine that may contribute to this effect seen in animal models. There's also the issue of timing after challenge with vaccination. And an example of correlation here would be respiratory syncytial virus, where you see immune enhancement in animal models depending on when the virus and the vaccination is the gap. To my knowledge, there is no evidence of disease enhancement from COVID convalescent plasma treatment in humans, even if they have low titer antibodies, which is suggestive that this is a theoretical risk rather than a practical risk. However, I think it's important to realize as we move forward in COVID vaccine clinical trials, these clinical trials have a follow-up of at least 2 years to follow up the volunteers in the studies to look for any potential safety issues or safety signals. Now, if we go to the pathway I showed you earlier, from basic science to preclinical to clinical to post-licensure, this is the typical pathway. When we actually move it into COVID, it's a very different pathway. We talk about not basic science, we talk about exploratory vaccines. When we talk about preclinical, we talk about preclinical in terms of we have to have manufacturing and a lot of vaccine being made already available to go into the clinic. And when we talk about implementation, we know that's going to happen. The issues come down to chemistry, manufacturing, and controls. I'm not going to talk about that at all because Roger's going to talk about that after me. So I'm going to talk about the exploratory, the preclinical, and the clinical. So where are we in terms of vaccine candidates? As of June the 20th, and I'm out of date, the number's probably higher now, there were 268 vaccine candidates that have been identified being researched in one country or another around the world. In addition, there's another 50 to 70 candidates being worked in academic labs and small biotechs. So there's a huge number of vaccine candidates out there at the present time. I think this is very reassuring because the likelihood of at least one or more of these candidates being successful as a vaccine, I think is pretty good. Even if you just rolled the dice and randomly took a guess, you would have at least 2 or 3 of them that are going to work. I think this is very encouraging. But of course, it's very hard to understand and evaluate over 300 vaccine candidates concurrently. In terms of the technologies that are being used for these vaccine candidates, these can be split into 9 different areas. We've got the traditional live attenuated vaccines using reverse genetics. We've got inactivated vaccine candidates, recombinant protein, peptide-based, RNA-based, DNA-based, replicating viral vector, non-replicating viral vector, and virus-like particles. So let's put some numbers next to these candidates. So overall, you can see there's many different vaccine candidates at each of the areas. The live attenuated, not surprisingly, has the smallest number 10 because the risk of attenuation virulence or virulence is difficult to judge. There's 11 inactivated vaccine candidates using a variety of adjuvants, yes or no. And then, as you can see, many recombinant DNA technology approaches being used with large numbers of candidates. You'll notice at the bottom of the slide, I've got 61 being unknown, and that's because the entities wish to keep their platform technology and their images confidential. And so there's a large number of these candidates for which we have very little information at the current time. So let's put this onto the vaccine development pathway. Currently in an exploratory phase, we've got over 175 candidates, plus 28 that haven't been disclosed what the imagens are. We have another 48 in preclinical manufacturing stage. We have at least 21 in clinical evaluation, and I'm going to talk about those 21 in a minute. Some people say more, some people say less, but my guesstimate is it's about 21 with a couple of others likely to go into clinical evaluation in the next week or 2. So I think it's important to say what we're doing in COVID vaccine research. The media portray it as an arms race to make a vaccine that gives the impression that the vaccine companies are all working in competition against each other to make a vaccine to make money. That's very, very incorrect. The race is to make a vaccine for a COVID before the next wave of disease. And it's an unusual situation that I can never, ever remember previously, where the vaccine development environment is very collaboratory. Everyone is sharing IP, everyone is sharing information. Everybody wants to control this pandemic. And it's really extraordinary the amount of collaboration that's taking place. And I think that's really important to say as compared to the view made by the media. However, everything is at warp speed, literally. And we can make a number of analogies with Star Trek, but boy, is it moving very fast and it changes day by day. It is really very hard to keep up with. There are multiple vaccine networks, as I would call them, a mixture of national and international networks that are coming together. One I think is important to talk about is the Accelerator. This is the access to COVID-19 tools, it's called the Accelerator. This is a partnership between nearly every entity you can think of, from the Global Alliance for Vaccine Initiative, the WHO, CEPI, Gates Foundation, Wellcome Trust, European Commission, every government, every NGO, private sector manufacturers. Everyone is working together to get these vaccines out to everybody who needs them around the world. The idea is to have a global equitable plan so that everybody gets vaccines around the world. It's not just the high-income countries that get the vaccines. There's an initiative called COVAX, where $18 billion is being put forward by a number of different entities to actually make 2 billion doses of licensed vaccine available to everybody equally around the world by the end of 2021. And the idea is that anybody who needs that vaccine will have access to it. And I think when you consider that we have over 8 billion people, if a quarter of those people have access to the vaccine in 1 year, I think that's a phenomenal move forward by everybody involved. Let's move into clinical trials. In terms of the clinical trials, I said earlier, we've got 21 vaccines in clinical evaluation, and I put a question mark because that's a guesstimate. Trying to get accurate figures from some of these countries is not straightforward. There are 2 DNA vaccines, 5 RNA vaccines, 5 inactivated vaccines, and these are all using the VeroCell platform, which is accessible to WHO, and they're all using beta-proprolactone for inactivation of the virus. There's 4 doing recombinant protein, 2 doing replicating viral vectors, 3 doing non-replicating viral vectors, and you will not be surprised to note that there is no live attenuated candidates currently in clinical evaluation. Put some names behind these. The DNA vaccines are made by Inovio, United States. Genexine is in Korea. Pfizer, we'll hear later from Roger. We have CureVac, Moderna, Imperial College, and a Chinese company all doing the RNA vaccines. All the inactivated vaccines come out of China. In terms of recombinant protein, we have 4, Novavax, which is this international, mostly US, 2 from China, and vaccine from Australia. We have 2 replicating viral vectors from China, and we have non-replicating viral vectors from the UK, Russia, and China. So it's a wide spectrum of vaccine technologies coming out of different parts of the world, demonstrating the true international nature of the vaccine research that's being undertaken at the moment in terms of getting into clinical evaluation. I just don't have the time to give you the details of how different companies work together. Some people are getting adjuvants to support other ones. Others are being manufactured by different companies to help everybody get there. It really is very much an international effort with everybody working together. To date, only 1 clinical trial has been published in a peer-reviewed journal, and that's the Ad5 platform from China that was published in Lancet. This platform was being used previously for an Ebola vaccine that was deployed in Africa. There are other vaccine papers coming out in preprints, including the Pfizer vaccine candidate phase 1 study that was published, and it was a preprint yesterday. But to date, the only study we have is one phase 1 in a peer-reviewed journal. So there really is a paucity of data available to be interpreted whether one vaccine is better than another one, but all the studies so far look very promising. There are a number of clinical trial networks. Here in the United States, we have one called the COVID-19 Prevention Network. This is a new network being made by the federal government, where they've taken different entities out of the BARDA and NIAID networks, the old HIV network, the VTEU networks, and put them to make a new network to do the clinical trials. In particular, we have a large number of phase 3 trials that will go on for the next 6 months plus, involving different vaccine candidates. And this is the network that's taken responsibility for evaluating those vaccines. This network is also doing drugs such as remdesivir, and is also doing some antibody studies as well. It's a large amount of work being done in a very short period of time. I'll talk more about that one in a minute. There's also WHO has what it's called the Solidarity Clinical Trials Network. And the idea here in terms of the vaccines is to compare multiple vaccines head to head in the same trial across different countries at the same time. This is proving to be a real challenge because, as you might expect, the regulators in different countries have different ways of looking at the vaccines, and coming to a consensus where the same approach and regulatory views will be taken for all the vaccine candidates in different countries is not straightforward. And to my knowledge, the Solidarity clinical trials for vaccines has not taken off yet. We actually have 2 licensed COVID vaccines. And when I say licensed, they're conditional licenses. They're both out of China. An activated vaccine has been given a conditional license for use by the Chinese military. And a second one based on the Ad5 vector vaccine has been given a limited 1-year conditional approval by the Chinese military as well. To my knowledge, these are the only vaccine candidates that have got as far as getting to a conditional license. In terms of the United States, the US FDA published a guidance document for COVID vaccines in development for the preclinical and clinical on June the 30th. And this document provides what the FDA feels as the pathway to take a vaccine through to get it through to licensure and to a BLA. I haven't read it in detail, there's just too much time happening day by day, but it really is what we need to actually help us know what the regulator in this country is looking for to get a vaccine license. So an interesting point we have to consider moving forward is how will we look at the duration of the immunity? In particular, if you remember what I said earlier about the veterinary vaccines at the beginning for coronaviruses not being very good, poor immunogens, and need lots of boosters. And the questions that will rise, I think, out of the clinical trials that will be really significant is how many doses of vaccine do we need to give immunity? How long will immunity last without a booster? Will immunity be protected? It's all very well making antibodies, but we're going to have to identify, well, what is that correlate of protection? What is that quality of the immunity which is needed to protect people from COVID infection? The FDA guidance document that was put out on June 30th says an expectation of a vaccine to prevent disease or disease, decrease its severity in at least 50% of the people vaccinated. Some people have said that's too low a bar, but in fact, if we had any vaccine, that would be an advantage over the current situation as we are in at the moment. I just want to finish by talking about the local institutions involved in COVID vaccine research. I think it's important to recognize there are a lot of organizations locally working in COVID vaccine research. In terms of vaccine candidates, Baylor College of Medicine, MD Anderson, Methodist, Texas A&M, and UTMB, and some local biotech companies all have candidates. I am not going to describe any of those candidates because I don't think that's my role. You need to approach those particular individuals. I just want to recognize there's a lot of research being done, and I apologize if I left anybody out. In terms of preclinical activities, there's a lot being done at my own institution at UTMB with the Galveston National Laboratory and its biocontainment capabilities. We have a preclinical testing group there, which is used to doing testing as part of a regulatory pathway. And we have extensive diagnostic vaccine support for high throughput neutralization assays. And with that respect, Pei-Yong Shi was one of the co-authors of the Pfizer preprint, where he developed neutralization assays that were used for high throughput screening. And again, I apologize if I left anybody out.
3 minutes, Alan.
Thank you very much. I'm nearly there, Stan.
I know.
So in terms of COVID vaccine clinical trials, to my knowledge, there are 3 entities locally that are doing it. That's Baylor College of Medicine, Texas A&M, and ourselves at UTMB. And again, I apologize if I left anybody out. I'd like to spend a few minutes concentrating on Baylor College of Medicine. You don't hear much about them in terms of their clinical trial capabilities, but they have an exceptional group there. They're one of the 9 NIAID-funded vaccine treatment evaluation units. Anna Sahali is the PI. It's been going for over 30 years. It has a great history of being involved in vaccine development and clinical testing, and a huge infrastructure. As you can see, they do respiratory disease, enterics, emerging infectious diseases, biodefense, a really large portfolio. Not surprisingly, they've gone into COVID. They're doing diagnostics for serology and PCR. They've been involved in the ACT studies, which are the adaptive COVID-19 treatment trials. This is what gave to the remdesivir as the current state of practice. And they do a number of surveillance studies. Probably critically important is they are part of the COVID-19 prevention network as part of Operation Warp Speed. And I can't talk about any candidates because I'm not allowed to, but essentially you're going to see a lot of phase 3 clinical trials take off starting in July. And I hypothetically would not be surprised if local institutions are involved in doing those clinical trials. And I should say that UTMB is one of the subcontractors as part of that group with the Baylor VTEU. So I just want to finish with, will we have a COVID-19 vaccine? I think the answer is yes. When will it be available? I think we'll have at least a first generation vaccine by the end of 2020. And I say first generation because who knows what vaccine candidates will come through next year. The ones we get this year may be great, we don't know. But I think we'll have at least what I would term a first generation vaccine. I think it's unlikely we'll be able to get 300 million doses to immunize everybody. And that assumes everybody only gets 1 dose of vaccine. I think it's far more optimistic and realistic to have a vaccine for everyone in the US during 2021. Will this vaccine be good? I don't know if we can answer that question. Hard to say at the moment because we still don't understand the correlate of protection. How many different vaccines will we need? My guess is probably 6, at least, if we're going to cover 8 billion people living on this planet. Thank you for listening to me, and I'll be happy to take questions at the end. Thank you.
Many thanks, Alan, for a warp speed tour of the vaccine landscape. We're going to hold questions for Alan till after Roger's talk. I'm delighted to introduce Roger. Alan, I had known for about 25 years. Roger, I think we're pushing 40 years now, which is hard to believe since you're like 35. So it's interesting. But Roger is the Vice President, Head of Global Chemistry, Manufacturing and Controls of Pfizer. He's in charge of a vast empire, I would say, looking at the global development, preparation, prosecution of Pfizer's regulatory and CMC development of new products, including vaccines. And that's why we're very delighted to have him join us today. He really is, has been very busy in regulatory development and CMC manufacturing for pharma, one of the leaders in developing quality by design, and have advocated really for global regulatory harmonization. He was awarded a few years back, Pharmaceutical Discovery, Development and Manufacturing Forum Award by the American Institute for Chemical Engineers, a highly prestigious award and well-deserving. He's been in pharma now for pretty much since he graduated from Carleton College a few years back. He's worked with Searle, Monsanto, Pharmacia, and he's now been with Pfizer for many, many years. Prior to that, he was a medicinal chemist. He has at least 24 patents under his belt. He's kind of given up bench work a while ago to take on much bigger responsibilities. So I'm going to turn this over to Roger. And Roger, again, just delighted that you could take time away from your very busy schedule.
Thanks, Dan. Can you hear me okay?
Yes, indeed.
And everybody should be able to see the presentation. Let me put it in presentation mode. There we go. So thank you very much, Stan. I appreciate being invited to present. As Alan mentioned, I'm going to focus predominantly on the quality end of putting together a development paradigm for the COVID vaccine, but I am not going to talk about it in terms of the specificity associated with Pfizer's initiative. And there are a number of reasons for that, and most of them are here in this disclaimer, or at least covered under this disclaimer. There is an awful lot of information, and it's changing very quickly. And as Alan alluded to this, I can't even keep up day to day because things are changing so fast. Yesterday, I was a bit surprised when I saw the following announcement from my own company, which indicated that we had some positive, preliminary positive results for the vaccine program that we are running. Not because I wasn't told about it in advance, I just didn't know when it was coming out. And interestingly enough, this is part of the speed or the warp speed that everybody's talking about that is forcing us to really move along with it in parallel rather than in doing anything in a sequential fashion. So what I'm going to try to do today in the next 25 to 30 minutes is just go through some of the challenges and some of the issues associated with developing a vaccine from a quality perspective and making sure that we have the appropriate level of quality assurance. I'm going to try to balance that with some of the regulatory expectations. And I want to say up front that I have tremendous respect for regulators. They don't always see things the same way the industry does or the way that developers of medicines do, but they are there to protect the public, and their questions and their engagement is critical to delivering high quality products and making sure there's quality assurance for all of our products. So I've been fortunate over the last 20 years, 25 years, to develop relationships with a lot of them to ensure that there's effective collaboration so we understand each other about what's needed and what needs to go forward in order to ensure that quality assurance. There are three pieces to this presentation. The first is I'm going to go very briefly over some conventional vaccine development. And this is based on my experience. It is not necessarily reflective of other companies or the industry as a whole. I will talk a little bit about the response to the COVID-19 program and the paradigm that it's creating, or the potential paradigm that it may be creating in a speculative sense. I will talk about it as what can we do to make sure that we can deliver this vaccine in a year, because frankly, this is a very unprecedented period of time to try to deliver a complicated medicine like a vaccine, particularly one of this nature. And then finally, I'll just talk very briefly about some of the global regulatory obligations that we have, because they tend to pose some of their own challenges sometimes, and it's useful to understand how those challenges fit into the overall paradigm. So for conventional development, Pfizer's been in the vaccine business not very long, frankly. When we bought Wyeth in 2010, we inherited their vaccine portfolio. And so for the last 10 years, many of us who have been getting educated about not only the technical and the scientific rationale for moving those vaccines forward, but also the development paradigm that is used in the regulatory requirements associated with that. Just as an example, and this is based on, like I said, probably the last 10 years as we've gone through development of our own vaccine portfolio, generally from phase 1 to approval of our applications for commercial distribution, it's taking between 4 and 6 years. It used to be closer to 10 to 12 years just for this period. And I think Alan mentioned it's about 20 years from discovery to actually getting the vaccine approved. So this section of that overall, what I would call a prolonged timeline is about half of it. And the development perspective is fairly significant in terms of all the attributes that have to occur. 2 reasons. One, companies like mine do not want to invest a huge amount of energy and a huge amount of money up front if the vaccine will fail or any drug will fail. Because frankly, a fairly large proportion of our budget pays for failures more than it does for success. And so as a result, we're trying to figure out better ways to hone that. And usually going into phase 1 and going to from preclinical into the clinical development is a significant step in getting us in that direction. Generally, vaccine development will require between 15 and 60,000 clinical subjects, depending on the type of vaccine and the type of therapeutic area. We normally do sequential development. And you will see in a few minutes that is different from what we will have to do with COVID or many of the other, what I would call accelerated programs that we're looking at for vaccines or any medicinal product. And we have to look globally. Most vaccines are initially approved in a given country, and then companies or institutions will try to get them approved in other countries in a sequential fashion. In this case, it will have to be parallel and global as much as possible for the most part. There are a number of steps through this. I'm not going to go through this, but basically we go from safety to dose ranging to efficacy and then into commercialization during this period. And we usually look at adults first, and then go into geriatric patients next before we go to adolescent or pediatric subjects. But that isn't always the rule. It depends on the type of vaccine. What I'm going to focus on is right here under quality assurance: characterization, control, consistency, and compatibility. Every product that Pfizer makes, every product that the industry makes must demonstrate that we have appropriate and complete characterization. We understand the control of the quality of that product. We manufacture consistently and reliably, and we have compatibility when we make changes. And we do make a lot of changes because we're trying to go from small scale to scale up. We try to modify and optimize dosage forms and processes to reduce the time that it takes to manufacture. In this particular case, vaccines in particular, and in particular the COVID vaccine, we are looking at characterization in terms of transcription, construction, and historical qualification. There are a lot of other components to this, but by and large, the information that we would provide to substantiate characterization would fall into these 2 buckets. For control, there are a number of areas where control occurs, and there is a misnomer among regulators and among a lot of industry folks that control is imparted by a specification. It is not. Control is managed through the process. The specification will provide us with confirmation that we have control. And that's really critical when we're looking at designing and developing a product, and in particular when we're moving very quickly, because we really do need to understand how that process is going to be reflected in the confirmation of control that we're trying to achieve. And it occurs across the board from the manufacturing processes all the way to when we have to have that drug administered with the instructions of how it's supposed to be administered and maintained in the field. With respect to consistency, there are essentially two areas, and they're both validation areas. Methods must be validated, and the process must be validated so that we can demonstrate that the consistency and reliability and sustainability of the product and the methods used to test that product are in place. And then finally, as I mentioned, the comparability, changes to manufacturing analytics occur throughout development, and there needs to be, over that course, a good comparability story so we can show a seamless connection from the beginning to the end. This is partly to ensure that patients who receive the vaccines and the data from those clinical trials is consistently aligned with what we expect to see commercially. Now, this is just a roadmap of a conventional product development picture, or I'll say timeline for quality. We usually in phase 1 and phase 2, we identify clinical performance characteristics. We end up ultimately defining the critical quality attributes of the product so that we have a design in place for the product. And then we shift to the process, and we determine from the process the critical process parameters that inform us about the quality of those critical quality attributes, whether or not they're being maintained. And this is an iterative process. Usually for a conventional process, we may go through 3, 4, 5, 6 sequences of this throughout the development paradigm before we get to the point where we are able to launch the product. And then after we launch the product and we have a significant amount of experience, then we develop a process capability understanding of PPK or CPK and go through continual improvement. So I'm going to move on to our response to the COVID-19 paradigm. In essence, what we are trying to do is truly reduce the amount of time, and you saw the time that I had, 4 to 6 years, down to 1 year, which is a huge, huge undertaking, and particularly with the level of capacity that is being required for us to develop. This is not a trivial matter, and it requires a fairly significant, well thought out approach that will likely have, fits and starts as we go forward, and we've already seen that since we've begun on our journey at Pfizer. And Alan referred to this as warp speed. I find it humorous to some extent that we're talking about warp speed, because in the end, as Alan also said, and I will emphasize, we must maintain the appropriate level of quality assurance. We cannot put a product out there that does not have that. And the regulators are keeping an eye on us, but we have an obligation before we even get to the regulators to ensure that that's true. So the challenge that we face is to identify, develop, and establish a reliable, robust, sustainable commercial manufacturing process and launch a large volume of vaccine doses globally within a year. And for our stakeholders, they're very excited, because if we can do this, obviously we may actually make a dent in the situation, the circumstances that we're facing right now in the world. But to development people like myself, this is a huge, huge concern. We are compressing the time for product design, development, regulatory review, and inspection. And we have to operate in a more parallel rather than sequential development, which is not something that we're used to doing for much of this, because we learned so much by doing many of the sequential activities. In this case, we're going to be doing a huge amount of work at risk and then confirming whether or not those risks are mitigated over time. The program objectives that I've got here are generally the program objectives that many companies or many groups that have been focusing on COVID have taken on. This comes from what Pfizer has given to me and to my group and the teams that I work with to ensure that we can do this correctly. So our first initiative was to assess the clinical outcomes of the 4 constructs that we currently have in the clinic. And that, in the announcement you saw, that has started to become part of what we are evaluating. Second, we have to deliver a commercially viable vaccine by the end of 2020. That means we have to have a commercially viable process that has the capacity to provide millions, if not billions, of vaccines over a period of time. We will move from phase 1 and 2 to phase 3 in parallel with regulatory applications for commercial approval. So in other words, we are in the middle of looking at the possibility of an investigational evaluation of the vaccines in a sizable number of subjects while we are preparing and submitting commercial applications for commercial approval. That is the plan anyway. And subsequently, we will manufacture 100 million doses by first quarter 2021. So no small task. I put this together because this is an example. If you look at these four sort of, I guess you'd call them supply volumes that arise from different approaches that could be taken. This is the projected sample strategy for each of those four. The green represents emergency use authorization, and I'll talk about that in a minute because there has been no vaccine that has been granted emergency use authorization since it was put in place by the FDA. And there may be an opportunity for one vaccine, I don't know which one, if they have the requisite data to be able to qualify for that, and I'll explain that in a minute. The blue line is a biologics license application for COVID-19 that may be used as a strategy going forward with a very reduced timeline. You're looking at about 3 months for approval before we can launch it commercially. The orange line is the normal, what we call breakthrough biologics license application under an accelerated program, which usually can be approved by the FDA in 4 months. And then this more purple line is reflective of what would be a traditional BLA, where we start manufacturing at risk during the review of the BLA. And just before we get approval, we ratchet it up because there's a fairly good likelihood at this point that we will have approval and we'll be able to move forward. And these are obviously the optimistic regulatory approval diamonds showing that. I want to go back to EUA because this is a provision that the FDA has offered if we have the requisite data to substantiate the safety and efficacy of the vaccine and the requisite quality, that we could start to administer it to more patients outside of the clinical trial and use that as the basis for developing a full-blown commercial application going forward. One of the other things that Alan mentioned and is really important is that there has been tremendous collaboration, much more so than I think I've seen for anything in my career with this product and with this therapeutic area and concern. The FDA and the EU in particular have been sharing a tremendous amount of information, and they are doing an awful lot to try during this pandemic to make it easier for themselves and for the industry in some of the requirements that we face, particularly inspections, and that's what this particular blurb is referring to. I've listed a number of guidances, one in particular that Alan showed, the development and licensure partner guidance. There's a partnership, that's where this came from. There are FAQs from FDA on EUA and the guide for EUA as well. I'm not going to spend much time on this because, quite frankly, I'm not sure how the probability of that is going to play out for this, but if it is probable, it is something that is useful and would give us a lot of opportunity to gather data more quickly. So the FDA expectations for CMC COVID-19 in the guideline and the guidance that Alan and I shared isn't much different from what we would have expected for a traditional vaccine. Quality cannot be compromised. But the way that regulators look at quality is in two pieces. One, the regulatory commitments that we make. Those are the descriptions, the specifications, the things that we make as commitments for the life cycle of the product, and the data that substantiates them or confirms that those descriptions and specifications are robust. And one of the areas where we believe, and I'm not alone in this, it's just that Pfizer has been relatively aggressive in pursuing, is for accelerated programs being able to provide, after we get approval, much of the confirmatory data beyond where the risk might be of a concern. So we're actually using more of a risk-based approach, and we think that that can be leveraged here for COVID-19. Where this plays out most significantly is in validation, where we're looking at process controls, method validation, and perhaps even drug product operations validation. In the current guideline that Alan and I referred to, this could be postponed. There is a statement that says it could be postponed for drug product operations, which does offer some opportunity to accelerate in drug product manufacturing. The other areas where we have some opportunities, but quite frankly, it's difficult for us to justify doing it, is in storage stability, container closure integrity, making sure we have the appropriate container closure and its integrity is maintained through the life cycle. and product expiry, and then also facilities and inspection readiness. And frankly, facilities and inspections really are at the heart of the foundation of quality. If you have robust and solid facilities, you will go through an inspection much easier, and it forms the basis for giving confidence to regulators in the quality of your processes and the quality of your product. Now, when regulators look at submissions, applications that are submitted, many of them will tell you, and I've heard this over the last 25 years or so that I've been in regulatory, that if we give them a high quality submission, the assessment time is very, very short, and that makes sense. If we put everything in there, there are no deficiencies, it's clear what we're doing and why. We've met all the criteria that they're interested in, it's not a problem. The trouble is, most of the time, and particularly in accelerated versions, it's not so easy to do that because there are going to be, from time to time, gaps in our information. And in this particular case, it is likely that there will be. And the real onus of responsibility is on us in the industry to figure out mechanisms to be able to provide either a plan or assurance that those gaps have a very low risk or probability of likelihood going forward. So where does that leave us? What direction do we take? How do we go forward with the COVID virus and deliver to the objectives that I mentioned a few minutes ago? Well, I have a suggestion, and the suggestion is a strategic one. I'm not sure that even my own company will agree with me that we will go in this direction, but this is something that I've thought of over the last few months as we've been putting together our plans, and I think it's useful to consider going forward. If you think about it, regulatory authorities are currently looking at investigational applications that include an awful lot of quality information. And we're going to be running those clinical trials with subjects of 1,000 or 15,000, up to 15,000 subjects or more. There is a good chance that the quality assurance demonstrated for those subjects could be extended to establish an acceptable preliminary and provisional commercial regulatory application, as long as we have sufficient demonstration of consistency in manufacture, consistency, reliability in manufacture, and robust product quality assurance in terms of our criteria for controlling the product. And there is a commitment by applicants to provide supplementary content reflecting the continuous improvement, verification, and confirmatory data going forward. We have used this in other programs that are not quite so complicated. And as I said before, it's a distinction between what we confirm up front and what we would confirm later. So putting together a robust discussion early on with some limited information that suggests that we do have an appropriate level of quality assurance, and then providing additional information to confirm that at a later date. And some of the examples of where this might occur, it goes back to what I talked about with respect to characterization, control, consistency, and comparability, are in areas where we will, if we follow this, have a limited amount of information in the manufacturing processes and specifications. And we will have to, through scale-up, site transfers, and manufacturing adjustments, and additional work on analytical criteria, including validation, have to provide that at a later date. So fit for purpose becomes the watchwords going forward, and we'll see if the regulators are moved to accept that for something of this nature. In addition, suitability of analytical method validation has often been accepted in the past, not quite so much for vaccines as it has for biologics and small molecules, but there have been appropriate plans, and there is an opportunity to leverage that. Because we will have a limited amount of experience, we would have PPQ and reliance on quality verification rather than formalized process validation, which I do think is appropriate and can justifiably demonstrate robust manufacturing. And then finally, analytical comparability with limited need to go back to characterization, because we believe that if we fully characterize this and the changes we make do not have a risk of impact, influencing a change in the character of the molecule, there is no need to repeat characterization. And so what we are doing at Pfizer, and I know a lot of companies are doing, is preparing to meet with the FDA and the EMA and other regulatory authorities around the world to talk about these topics. And I'm not going to go into detail with these topics, but the primary one, the one that's at the very top, is the product control strategy. Do we have the requisite information and the quality in our process, in our manufacturing, and can we confirm it with our specification criteria to adequately say that we control that product to meet safety and efficacy expectations? And these are the things that we would put in a commercial application, and we would supplement in post-approval evaluation if the speed of this continues to move at the pace that it has. Likewise, for GMP and inspection criteria, there are a number of areas where we are requesting, and we will be requesting FDA to engage us on these as well. So let me talk just briefly about global obligations, global regulatory obligations. As I said, I've been in the regulatory gig for a while, about 25 years, and I've seen a tremendous increase in the interest in companies moving into other countries and working with other regulatory authorities in other countries to try to understand better where we will put our emphasis in terms of our investment in development, and then subsequently try to modify whatever we've done to accommodate certain regulatory authorities. Every regulatory authority that comes from a sovereign nation feels special. They have an obligation to their population within their country to ensure the quality of the products are maintained. As a result, they sometimes have what I would call divergent expectations. Regardless of the science that is used to justify the product's quality, there are nuances, and a significant number of nuances, and I'll talk a little bit about what that is. So when we talk about global considerations, first and foremost, we have to talk about the supply chain capacity, because obviously this is huge. And the numbers that I've seen for trying to manufacture vaccine candidates for COVID right now are well above 2 billion to 3 billion within the next year, year and a half. And that's significant. And frankly, the overall capacity that the industry has may not be able to do that easily without some changes to some of the manufacturing sites, and maybe even adding manufacturing sites and working together. I mentioned divergent regulatory expectations. For any plan to work and to move quickly globally, regulators will have to be working together. They will have to be collaborating not only with the industry, but with them amongst themselves to ensure that we can get approvals within those countries in similar fashion and around the same time, or in simultaneous fashion, in order to be able to deliver the product. Redundant testing requirements are frequently a feature of commercial expectations in many countries. Europe requires it, China requires it. If you manufacture a vaccine in the US, you have an obligation to ensure that FDA sees your certificate of analysis so that you can release the product for most vaccines. And so in order to deal with redundant testing requirements, there has to be one standard that we have to use, and perhaps look for opportunities to waive this requirement, at least initially, as a provisional expectation. There are important export boundary issues. We've already seen them with some of the vaccines that we are working on, getting vaccines between ourselves and our partners. BioNTech is a German company, and we have multiple sites that are involved. And so moving those things back and forth has been a bit of a challenge, particularly during this pandemic. And then distribution challenges. This vaccine right now has to be stored, one of the ones that we're looking at a very low temperature. We're hoping to be able to raise that temperature at some point, but anytime you have cold chain supply, you have a significant amount of extra work to evaluate and make sure that distribution is not compromised. So in summary, in response to the urgency of the pandemic, the rapid development has been really a challenge because of the extreme fluid nature of the information that's available. As Alan mentioned, and you know, I've dealt with it as well, things are changing by the hour in many cases. We're making decisions that are significant and have ramifications for how we're going to develop and ultimately manufacture our product, in addition to the unprecedented scale. As I mentioned, collaboration is absolutely critical, particularly between government agencies and the industry. And while we want to make sure we have consistent, reliable, and robust product quality assurance, opportunities for flexibility need to be leveraged at this point in order to be able to meet the rapid expectations. And then finally, the alignment among the regulatory authorities with respect to some of the expectations for approval and for moving these products forward is critical as well. Don't know whether or not we're going to end up seeing this vaccine as something that will be an annual vaccine. Somebody made a joke about this yesterday, and it was a bit of gallows humor for us since we're not even sure what we're going to have for the initial vaccine. But nevertheless, it speaks to some of the uncertainty that we're facing overall. And with that, I'll say thank you, and I guess it's time for questions.
Well, very good. Thank you, Roger. Once again, I really, really appreciate it for giving us a good view of what we're facing as you and your teams around the world, look to get a vaccine, which isn't approved yet, but as we hope to get it approved, what it's going to take to get that into the waiting public. So the way we're going to handle questions typically is through the chat, and if you do have a question, if you could pose it in the chat bar, you all should have access to that. If it's for someone in particular, please let us know. I'll try my best to moderate these without screwing it up too badly. If you have any comments, any suggestions, any praise for our panelists, for our speakers, I'll also pass that on. I see something from Wesley Sundquist, I'm not sure, who just said wonderful presentations. I think he is referring to both of you, and I echo that. So again, wonderful job. A question, we'll start off one for Roger. This is from Clark. So his question is, from the time a vaccine is shown to be effective, how long would you estimate to produce, let's say, 300 million doses? Best case, obviously worst case is never, but best case, how would you estimate that?
That's a great question. There are two ways to kind of address that. One is we are planning, and I think most companies are planning, most groups are planning to manufacture risk even before we show efficacy. I think I tried to show some of that in what I was presenting. That box was really an approval box, a period during which you might have an application in front of a regulator. But frankly, we're doing that in parallel while we're actually running the clinical trials at this point, or we're planning to. So we would have a modicum of product available, hopefully in the, I would say, tens of millions, fairly well along before we get to the point where we've demonstrated what would be considered efficacy. And then we would ramp up with additional capacity immediately thereafter. My guess, depending on some of the regulatory constraints, it could be anywhere from 3 to 4 months, but that's a guess. I think there is some capacity. I know we have some capacity, and we're working with other companies to ensure we have additional capacity in the event that we need to do that.
And his second part of Clark's question is, any thoughts on how you're going to manage, let's say your company or other companies might manage the idea of US versus the world distribution for vaccines, right? I mean, yeah.
It's a really, it's a difficult question to answer. I think all of us would love to be able to say that we would just continue, it's like Doritos. We're just going to keep making more and more of them and be able to distribute them as we get an agreement and approval with regulatory authorities around the world. In fact, it's not quite that simple. And there is an awful lot of politics behind this that I'm not going to get into that tends to drive, how companies look at this. The good thing is our partner is in Europe, and we have agreed with our partner that we have to be globally focused. So at least in principle, that is the direction we're going to take. But a lot will depend on what the regulators and the politicians, to some extent, in each of those markets will determine, and how quickly they'll determine whether or not we can launch products.
Very good. Here's a question from Alicio. I may have screwed up the name. I apologize. And this is for both Alan and Roger. They would like to know, are you afraid that perhaps we're moving too fast without knowing the pathology and pathogenesis of the virus, which may be variable?
I guess that's one for me probably first. I don't think so. There's a number of working groups that are looking at disease enhancement, and that's happening concurrently with all we've talked about today. And there's obviously a risk out there, but everybody is aware of it, and everybody is conscious of what needs to be done to analyze that, particularly when it comes to the clinical trials as well as the animal work. A number of countries have working groups that work in parallel with the work that's going on by the companies. As we sort of said, it's warp speed, but as we both said, everything is being done as it would with a normal vaccine. It's just everything is happening concurrently. And you could argue we don't know enough about the pathogenesis. I think you could argue that the animal models don't recapitulate human disease as well as we would like, but they're what we have at the moment. And everybody is well aware of the potential of disease enhancement. And as I said, there are a lot of working groups set up by a number of countries and international organizations to monitor this in real time as we go forward. As I was saying, everybody's working together.
The only thing I'd add is I think for most of the candidates, the safety of these vaccines will be substantiated early on. And so at the very least, you know, we may see some issues with some of the vaccines in the clinic. I think that'll be an early watch out if there are any concerns. I am nervous about putting things out there as quickly as we are. I know how complicated this is, you know, and having been in the industry almost 40 years, I've seen things happen. On the other hand, I've never seen such a significant amount of commitment and collaboration to try to ameliorate where the risks exist. And so as a result, I have a lot of confidence that, you know, we're going to deliver something, and we're going to deliver it maybe not by the end of the year, but hopefully not much further beyond that, if everything works well.
Very good. We have a question from Rebecca, and this possibly more for Roger. She said, let's assume you've begun manufacturing and distribution, and if there was a change in something like culture media that results in producing substantially larger amounts of vaccine components, what would such a change mean from a regulatory standpoint? When would you introduce this change into the regulatory cycle? So a significant change in manufacturing, how do you handle that?
Yeah, the first thing we do is we have to, through comparability, we have to demonstrate that it hasn't had a deleterious impact on the product. And anything that is related to improving the speed of delivery is clearly something we want, because it'll make it obviously we'll get the product to patients quicker. I think the comparability exercise is something that has to be negotiated, and each regulatory body has a different view of what that may entail. You may have noticed on one of my slides, I mentioned in vivo evaluation. Sometimes that in vivo evaluation can be animal models if they're robust enough. In other cases, it's actually introducing, if it's a significant change, introducing that into subjects to demonstrate that there has been, you know, little or no change in the comparability of the product. So it really is dependent on that. I would hope we could use analytical comparability for almost everything, or as much as possible. But again, it assumes our analytical methods are robust and are capable of detecting, you know, permutations.
And Stuart has a nice question that both of you could jump in on. It seems like as the epidemiology of the infection seems to be not necessarily changing, but at least looking more and more, shall we say, lethal for elderly individuals, how do you anticipate that's going to modify the clinical trials? Are you going to start focusing on particular comorbid patients, elderly patients, et cetera? What are your thoughts on how that should progress?
Okay, I guess I got that first. Stuart, I'm sorry, that's a difficult one to answer in the public domain. Each of the vaccine candidates has their plans for their trials for particular age groups and the type of groups of people they're looking for. Unfortunately, that's not in the public domain yet. I think it will happen soon when the phase 3 start in this country. But at the moment, that information is confidential, I'm afraid. I get asked it every day by my friends. I'm a particular age, will I be allowed to be included in the trial? And the answer is, I'm afraid, that is not allowed to be answered until it's in the public domain, I'm afraid.
Yeah. I have nothing to add, and that's exactly right. We're not at liberty to share that.
Okay. Question from John mostly to Alan. It seems as though the federal government has made a big financial bet on just a few companies. We learned today that there are about 250 vaccines under development, but without funding, how many of these will truly have a shot on goal? Thoughts, Alan?
Yeah, so John, I think that's a complicated question to answer. Most of the information that we get in the public domain comes from the media, and the media don't give the whole story. And you hear about a small number of companies that are being done in Operation Warp Speed, but you in different venues, you hear different entities being mentioned. And you have to remember that many of these vaccines have been evaluated in multiple countries concurrently. And I don't think it's just a small number being analyzed as you indicate there. It's a larger number. It's just that it's not in the public domain the way it's presented by the media. It's unfortunate, but most of these press releases are the way you get the information, not from accurate information. Like, for example, it's very hard to get hold of the actual documents for Operation Warp Speed. You can't really get them easily to find to read what the process is about. And John, it's very hard. I mean, it's just that not everything is not presented, and it's not the fault of everybody really. It's the fact that everybody's so busy, there's not enough time to get the information out. And of course the media will sell newspapers, and they just want to get their snapshot out for the next day, and so they pick a certain part of the story to sell it and ignore the rest of it.
Nothing to add. Well said.
So Juan has a question to both of you, and this might be more of a philosophical question. Sorry for ad-libbing on there, but the question is, when the vaccine becomes available, but what happens if 1/3 of US citizens refuse to take the vaccine? What will happen to the whole society?
I have no idea. We're doomed. How's that?
I don't think that's going to happen.
Let that out, Raj.
I agree with Alan. I don't think that's going to happen either.
Humans are selfish animals, and they make a risk assessment. They make a risk assessment not to take a flu vaccine or a measles vaccine because there's not much disease. Humans will make the risk assessment. If there's a lot of disease around in the fall, there'll be a queue to get the vaccine. If there's not much disease, they'll make the risk assessment not to get the vaccine. It's the society we live in, I'm afraid.
Yeah. You know, it goes back to the question before about wanting to participate in the trials. Everybody at Pfizer is disqualified because we can't introduce any bias. But everyone at Pfizer wants to be in the trials. That's 90,000 people right there. So we may not be a representative population, but we're a significant population. And I think that's true of many places. The town I live in is small, but I've heard from at least about 2/3 of the people that I've talked to who would gladly participate in a clinical trial and definitely want a vaccine.
This is a question from Mel. Is it known what the distribution of recombinant spike protein sources are relative to the degree of glycosylation of the protein in the vaccine candidates? One more time. You got that one, Alan?
Yeah, I'm trying to read it. Distribution. All right. When you say the sources, do you mean the manufacturing sources of how the proteins are made differently, or is that a more genetic, phylogenetic question? So glycosylation doesn't really cover the main neutralization sites that we've been looking at the moment. It makes a difference to the structure. Is that what the question was getting at?
I think so. I think depending
on...
on...
RBD has no glycosylation on it. There's glycosylation in other parts of the protein, but the RBD is not affected by it, if that's what the question meant. Phylogenetically, clearly the virus does change, and there'll be differences in glycosylation also, depending on what cells it's produced in.
And then Emmanuel has a question that I think really requires both of you to put on your very forwardly thinking hats, which you both, I know, have, is... So given this unprecedented experience, do you think it's paving the way to different methods of developing cutting edge medicines as you look to the future?
Alan, do you want to go first?
Thanks, Roger. As I said at the beginning, I'm involved in CEPI, and CEPI was set up the idea of taking modern technologies and being able to produce vaccines for emerging diseases and novel threats, disease X as we call it, at short notice. And I think it's happened far quicker than we anticipated. We weren't ready for it, but this really is the way we're going to evaluate all these novel technologies. Can they deliver in terms of their promise of plug and play? Be it RNA, DNA, or vectors, we're going to find out whether they can deliver or not. And we will probably know within the next couple of years. I don't think we'll know much earlier than that, because it may be depending on the dosages. And it may end up being heterologous prime boost. We'll have to wait and find out. But it's an active experiment in some ways. And we will know whether these technologies have the capabilities of delivering or not.
I agree. I think there are other technologies too that are actually helping to usher this along in the same fashion. Gene therapies in particular, that as a new modality is paving a different direction, and the speed with which many of the companies are trying to develop those products for rare diseases in particular is fairly significant considering the limited number of patients. And it is creating a new normal, or a better normal perhaps. I was at a meeting earlier this year at the National Academy of Sciences where a couple of folks from FDA mentioned better normal or new normal in terms of some of the things that I mentioned in the presentation I gave today, which is, can we provide the requisite information to regulators so that they can make a risk-based evaluation on a limited amount of data, because there is so much background information that they would be aware of, relatively speaking, on platforms. And many companies are moving to platforms. So I do think that this will help us get to a new normal and perhaps reduce the amount of time it takes to get from discovery to approval and get it to patients sooner. And my hope is that that will be for everybody's benefit, the industries, the patients, and everyone else.
I would agree.
And a follow on to that question would be, again, to both of you, again, looking, putting on your visionary hats, what innovations, resources, institutions would you actually like to see established from this current pandemic in order to allow us to handle, in a better sense, the next pandemic when it comes? And again, Alan, you know, looking back at the 21 years ago when you were part of the dream team, right? Handling those pandemic, where are we going to be, how are we going to be doing, what do you want for us to handle it better 20 years from now?
I think there's reality and there's an ideal world.
Mm-hmm.
The reality is that politicians do knee jerks. If there's a Zika outbreak, we dump $2 or $3 billion in that year, then it's gone away next year, and unfortunately we forget it. The same happened with Ebola in 2016, 2017. We as humans, we don't learn very well that we need to make long-term investments. We can't just make a short-term investment and expect scientists to produce a vaccine eureka in a year, which is the way we tend to operate. That's a reality. I think somehow, if moving forward, we have to come up with a plan for long-term scientific advances with a fair amount of consistent investment so that we can actually get there at the end. And if you just look for emerging diseases, money gets cut from the surveillance programs. And so then we're surprised when something reappears and when we weren't prepared for it. And it's this long-term investment where you may go a few years where you get no return on investment. And people say, well, I want to save my money. But then next year, something big happens and we weren't prepared for it. And there's the challenge. How do we do that within the reality of the politics versus long-term science?
Yeah, and I'll take a bit of a selfish industry view because I work in industry, and also because I've been involved in a lot of innovations, technological innovations like continuous processing and manufacturing. There has to be an incentive for industry to invest, particularly private industry. The government will invest to some extent, but they won't be able to develop to the extent that industry can, because the industry has, frankly, the capacity to do it much better. So that finding that right balance, and one of the best incentives is the ability to work with regulators in a way, and other companies, to deliver that sooner and apply it sooner so that you can deliver products more quickly and more cheaply, as a matter of fact. So we've got to figure out a mechanism to do that. But right now, money dictates almost everything in the world. And in the pharmaceutical industry, whether we like it or not, It invades even the regulatory nature of what we have to face from time to time. Last year, I was in China discussing regulations that had just been issued in that country. And the reason they issue some very stringent regulations is because they're trying to bring up the level of development and control within their industry internally. They have 10,000 pharmaceutical companies in China. So they apply that across the board to companies that already have very good quality records. And that by raising that bar, it reduces our incentive to spend time and effort trying to get products into that country. So part of the discussion was, can we figure out an incentive-based approach for not only the companies within China, but for the multinationals to be able to deliver on that innovative approach?
Back to the chats, Shatong asks that they're wondering if a universal corona vaccine makes sense and is feasible. Yes.
It's already started. There's already groups working on universal corona vaccines, as there are with the influenza vaccines, with the same idea of having, you've got the structure function of the spike protein. Now you can work towards a universal vaccine. That's started already. But it's way back. I mean, it's not on the front line that we're talking about today, but there are studies that have been started doing that.
And Wong asks, once a vaccine is available, do you think it will bring any benefits to those that have already been infected and may still have circulating antibodies?
The quick answer, I think, is yes. The unknown is we don't know how long a vaccine's going to give immunity for before we have waning immunity. If you take the scenarios for MERS and with SARS-CoV-2, somebody who has a wild type infection, probably 6 to 9 months later, they will have waning immunity. And so, yes, if people have an infection now, if they get a boost with a vaccine, that will probably help them in the future, yes.
And then taking some of the questions that were submitted in advance and some additional ones. So do you feel there may be countries that might be more amenable to achieving kind of the equivalent of the FDA Emergency Use Authorization Act? In other words, that would say, yes, we're going to have a very low bar for you to start distributing the vaccine in our country because we really want to get through this economic nightmare as quick as possible. What are your, one, do you think that's going to happen? And two, if it does, what are your thoughts on that?
It already is happening to some extent from my experience with a few countries. One country that approached our company and talked to us at length was Brazil, Anvisa in Brazil, the regulatory authority there. They had on their books, I think, what ostensibly was a very preliminary EUA approach that they wanted to be able to use. And so they wanted to get some feedback from a number of companies. And so I think they were soliciting information. I do think, particularly if we're not going to get we're not going to get this drug out to every country at the same time in the same way, clearly. And if there are multiple vaccines, it'll probably be a hodgepodge. So my guess is that countries that don't get it as soon as the others are going to lobby very hard to try to do something like that. And that would be part of the discussion. How do we do it? Now, my preference would be, I don't know what Alan thinks about this, but I've seen it work, mutual recognition or mutual reliance, where a regulatory authority from one country will mutually recognize the approval from another country for the same drug. That would make our lives easier because then we would be able to, essentially in almost a simultaneous fashion, assuming we have capacity, be able to deliver product to multiple countries simultaneously.
I would agree with what Roger said. I think it's important to realize that the FDA doesn't have the lowest bar compared to some of the other countries. And I don't want to name them. Probably people don't realize in the background, when any vaccine is going to license it internationally, the regulators do get together and discuss how that will be released in different countries to come to some sort of harmonization if they can. There's a thing called the Developing Countries Vaccine Regulators Network, which has EMA and FDA representation, plus low-income and medium-income countries. And I've been involved in a few vaccines where we've had multiple regulators from different countries as observers, shall we say. of discussions at WHO to their involvement, so they know what's going forward in terms of how that vaccine would be recommended by WHO and then how it would be implemented. Countries don't work in isolation. However, each country has its own criteria and approval process.
And we have time. We're going to take one more question from the chat on the side. And as we take it, again, I just want to thank both of our speakers for a great presentation. We appreciate your time. We know you're both, swamped with trying to get the vaccines developed and out the door. So from Upendra, what would you both view as a good readout of a vaccine success? How would you qualify a vaccine success?
Roger, you can do this one first.
Well, you saw the FDA's view of efficacy success of 50%. I think for me, it's going to be incremental. I think if we can get something out the door that has some limited efficacy of some sort, 50%, okay, I'd be happy with that for now. And then we'll continue to develop versions of that vaccine or other vaccines to make it better. I think because we're moving so fast, the pace is so great, we can't hope for anything else. We're not going to get perfection. There's no way, not at this point. We don't know enough, and the uncertainty is going to overwhelm us. So I think, let's take some, I'll call them baby steps, but quick baby steps to get there.
I agree totally what Roger says. The regulators have got more user-friendly, I think would be a way of putting things. For example, the malaria vaccine, it was 39–40% efficacy, and there was still interest in giving conditional licensure. And I think the regulators realize there's a need for these vaccines and are willing to help push it down the line. But Roger says, ultimately, we want a vaccine that's going to be perfect, up in the 80s, 90% efficacy. But that's unlikely we're going to get that sort of data in 2020 or 2021. It's long term. And as I said in my presentation, we normally follow up volunteers in these trials for at least 2 years. Just look at their immunity, how long it lasted for, when it wanes, are there any side effects? This is a long-term project. We're looking at a snapshot in 2020. But even if these trials finish enrolling as they're supposed to in the end of July, start of August, we're still going to follow them up for 2 years at least. So this is not a short-term project.
And on that, I think we're going to thank again the speakers, thank Dr. Tomlinson for organizing this. And all I will say is everyone out there, stay safe, stay healthy, keep your sanity. This too, we will get through. All right, so again, thanks everyone.
Thank you for the invitation.
Yep, thank you.
Thanks, Roger.
Thank you, Alan. Cheers.